about the protein synthesis today. So it is, you can say, well known as the, also known as the translation. So as you all know that DNA and genes, so the DNA is present in the chromosomes, or you can say, well, the chromosomes is made up of the nucleic acid or the DNA, and the DNA that contains the genes, and these are the sequences of the nucleotides, and these genes, they code for a protein that we known as the polypeptides. And these proteins, that's why they are important, as you all know that these proteins, they are used to build cells, and they do much of the work inside our cells or the body. So the proteins, they are made up of amino acids linked together by the peptide bonds, and though commonly you can say, well, there are 20 amino acids, and you can say a few years before there are two more amino acids, they are decoded, or that is the selenocysteine and pyrrolysines, but these are the rare amino acids. And so we have 20 different amino acids, and this is the structure of an amino acid. So you can have a centrally located carbon, and the side chains on one side, you can say amino group as well as the carboxylic acid group. And this is the polypeptide. So you can say the chain of amino acids is called as the polypeptides. So how this process of the translation begins? So as you all know that DNA is found inside the nucleus. The proteins, you all know that they are made in the cytoplasm of the cells by the organelles called as the ribosomes, and the ribosomes, they are available in the two states, that is free as well as the attached to the endoplasmic reticulum. If it is attached, it is called as the rough endoplasmic reticulum. So for the protein synthesis to begin, that the DNA's code, it must be copied and it taken to the cytoplasm, and in the cytoplasm this code, it must be read, so the amino acids, they can assemble and forms the polypeptides or the proteins, and this process is called as the protein synthesis. So for the protein synthesis begin, the RNA is the one of the entity, and because the mRNA is going to bind to the ribosomes, and you can say this is the comes from the nucleus. So together we can say, well, the DNA is the master planner of the protein synthesis, and the RNA is the blueprint of that master plan. And this is just to show, well, what is the basic difference between the RNA and the DNA. So the DNA has a ribose sugar. The DNA has a deoxyribose sugar, and the RNA has the ribose sugar, that is in terms of the pentose. And like the RNA, the other differences, you can say the RNA contains the uracil, and the DNA has the thymine instead of this. RNA is single stranded and the DNA is double stranded molecule. So this is the ribbon-like structures of the DNA double helix, and this is the structure of the RNA, is single stranded. And these are the some of the commonly found, like, or the types of the RNA, the mRNA, rRNA as well as the tRNA. So the mRNA, it copies the DNA code and it carries the genetic informations to the ribosomes. rRNA, it, you can say, along with the protein, it makes up the ribosomes, and the tRNA, it transfers the amino acid to the ribosomes, where the proteins, they are being synthesized. So mRNA is more, we are focused on the mRNA because it is the main entity, that is mRNA, though it's the tRNA, mRNA, both they are the main entities for the protein synthesis. So the mRNA is, you can say, it's a long straight chain of the nucleotides made in the nucleus, and it copies the DNA and leaves through the nuclear pore, and it contains the nitrogen bases, that is the A, G, C and U, and there is no thymine in this. It carries the informations for a specific protein, and it made up of 500 to 1000 nucleotides, and the sequence of these three bases or nucleotide bases in the mRNA is called as the codon. For example, AUG, it codes for the methionine or the start codon, and the UAA, UAG or UG, they are the stop codons. This is the structure of the ribosomal RNA. This is a single stranded made up of 100 to 3000 nucleotides. It's globular in shape. Also, like the mRNA, it is made inside the nucleus, and it associates with the proteins to form the ribosomes, where the sites of the protein synthesis. Then the genetic code. So a codon designate an amino acids, and an amino acids may have more than one codon. So that's why the 20 amino acids, or you can say 22 amino acids, and having the 64 possible codons. So a codon specify amino acid, and an amino acid coded by more than one codon. So this is the table of the all 64 the codons that is present. Then is the tRNA. tRNA is like clover leaf shapes, is a single stranded molecules that having the attachment sites of the one ends, that like for the amino acid, and the opposite end it has a nucleotide basis that is called as the anticodon. So the codon and anticodons, they are complementary to each other. So you can say the three bases of an codons, they are complementary to three bases of the codons. So this is for examples, the ACU, it is code for the mRNA, it is the codon regions, and it is complementary to in the tRNA is the UGA. Then before the mRNA, it leaves from the nucleus for the protein synthesis, it should be processed. So that is called as the processing and the RNA editing. So the after the DNA is transcribed into the RNA, the editing is done to make this RNA functional. That is the, in the editing you can say the introns, they are non-functional part of the DNA, they are the spliced out of the chain, and the exons, they joined to form, or they are joined to form a mature mRNA, and they are joined with the help of the ligase enzymes, and a guanine triphosphate cap is added to the 5' end of this newly copied mRNA, and a poly-A tail is added to the 3' end of this RNA. And this newly processed mRNA, it can then leave the nucleus for the protein synthesis. This is the result of, you can say, the processing. If you can see, well, these are the DNA having the non-coding regions in between the introns, and after the transcriptions, so you have the nascent mRNA, and then these introns, they are cut out, and the coding regions, they are joined together to form the mature mRNA, and then mature mRNA, you have a cap as well as a poly-A tail is joined, and this transcript or the mature mRNA, now it is ready for the protein synthesis. So you can say this mRNA transcript, then it's leave the nucleus through the spore, and it goes to the cytoplasm and attached to the ribosomes. So you can say, well, this process of translation, it is the decoding the mRNA into a polypeptide chains, and the ribosomes, it read mRNA 3 bases or one codon at a time and construct the proteins. So you can say, well, this is double helix of the DNA, transcribe mRNA, then this mRNA is coding to have the these polypeptides, and these polypeptides, they are, for example, forming the arginine, tyrosine, tryptophan, different amino acids, and they joined to, these amino acids joined to form a polypeptides or the protein. So one more things before this, like for the, as we all know that the protein synthesis, it begins or it happens into the ribosome. So the ribosomes is an important entity. So and the ribosomes, they are having two units, large and small units, and it is majorly, you can say, composed of 40% of the rRNA and 60% of the protein, and they have the two sites for the tRNA attachments, that is P site and A site. And it's also have one more site, that is E site, called as the exit site. So the first steps of the translation, that is the initiation of the protein synthesis. So the mRNA transcript having the with the start codon AUG, it attached to the ribosome subunit, and then this small subunit attached to the ribosomes. So that forms a initiation complex. So this is to show the first the mRNA molecules that comes to the small subunit, attached to the small subunit, and then the large subunit attached to the small subunit to make a initiation complex. And if you can say, well, this is a little bit in detail, that these large and small subunits, they are normally joined, but when the protein synthesis begins, there is an initiation factor IF3, it bounds to the small subunit, and this binding of this initiation factor preventing the large subunit to bounce to this small subunit, and that's why the small subunit is now available to the binding with the mRNA, and then mRNA it attached to the ribosomes. Then after this, then you have the first charged tRNA molecules with, say, like 4-methyl and 4-methyl methionine. It forms a complex with initiation factor 2 and GTP, and attach. Then the initiation factor 1, it bonds with this complex, and this is, so the first charged tRNA along with the initiation IF1, 2 and 3, all these together, this is forms as a initiation complex. So after binding of this, or when this complex is formed, all the initiation factors like initiation factor 1, 2 and 3, they are dissociated from this complex, and the GTP is hydrolyzed into the GDP and inorganic phosphate, and because of this this initiation factor 3, it preventing the binding of large subunit. When it is dissociated from the initiation complex, the large subunit comes and bonds to this initiation complex and forms a complete, or the 70S initiation complex, and it is the first steps, that is the initiation of the translation. So after this, the second steps is the elongations. So the ribosomes moves like the two tRNA with their amino acids. So the tRNA molecules with amino acid attached, they comes to the A sites, and the peptide bond formations between the two amino acids that the tRNA is carrying takes place. And then let's show, like this is the initiation of the complex. As you know that this is the first amino acid, it's bounded to this. Then there is a hydrogen bond between the codon and anticodons takes place. Then the second, the amino acids, or the tRNA with the second amino acid, it comes and occupies these sites, and there is a peptide bond formation takes place between these two amino acids. Then after this, the first, as the peptide bond formation takes place, so this second tRNA, then it has the two amino acids bound, and the first the tRNA, then it is leave through the E sites, and the ribosomes moves. So this tRNA along with this two amino acid, it comes to the sites, and then the A sites is free for the or available for the third tRNA having the third amino acids, and the same process is keeps on until the, so like this is the fourth tRNA comes, then the fifth tRNA comes, and so on. And so a little bit more detail about this initiation as the elongation though. So when this first initiation complex forms, then for the second, when the second charged tRNA comes, so it requires a elongation factor Tu as well as the GTP. They makes a complex, and then it enters into the A sites, and after this the charged tRNA is placed into the A sites, this GTP is cleaved, and this elongation factor Tu, it is dissociated from this complex, and this is then another elongation factor Ts. It helps in the regenerating this GDP or convert into GTP, that is again available for the next charged tRNA. So this thing happens, then the peptide bond formation takes place between the two charge between the amino acid of the two tRNAs, and then as the ribosomes moves. So when the ribosomes moves down to the next codon, that is the translocation of the ribosomes. So there is another elongation factor G as well as the energy GTP is required, and then when it translocates this the first charged tRNA, it leaves through or exit through the E sites, and now the A site is again open for ready to receive the another tRNA. So that is the second steps, that is the elongation of this, and after this third step is the termination. So the end product of the protein synthesis is the primary structure of a protein and a sequence of amino acids, that is you can say bonded together by the peptide bond. So like this, and so if you see, they summarize this is the mRNA, then you have this proteins, and this is the primary structure of the protein. This is the last steps, that is the termination of this the protein synthesis. So when this ribosomes moves over a code, and that is the terminator codon comes like a stop codons, like the UAG. So there is no tRNA that having a complementary to this stop codons. So this A site is then empty. Then there is two releasing factor RF1 and RF3, they comes and attach. So the RF1, it occupies this A site, and RF3, it forms a complex with GDP and bounds to this ribosomes, and that release, the polypeptide is released from the tRNA into the that present in the P sites, and after the release of the polypeptide, this GTP, it associated with the releasing factor is hydrolyzed to GDP, and then all complex, the tRNA, mRNA and the release factors, they are released from the ribosomes. That leads to the termination of the translation. Thank you.